It's very rare in drug development that you actually get to bring forward a new mechanism that has the potential to transform therapy for so many patients. And that's the case for Aerpio. We have first-in-class treatments available for diabetic eye disease, particularly diabetic retinopathy and potentially DME. We have a pipeline of drugs, a small molecule and a large molecule activator of Tie2. We believe the small molecule drug, which has phase 2 data ongoing, a phase 2 trial ongoing, is the least invasive and potentially the most effective way to activate Tie2. We have demonstrated activity in a phase 2 trial, which I'll show you today. And obviously, we're taking it into the diabetic retinopathy arena where the market potential is really huge. We do own all the global rights to all our molecules and the approaches we've taken, and we have an experienced management team and board. Now, when you do a new mechanism, or actually when you do any mechanism in drug development, it's really important to ask two fundamental questions. Does your mechanism operate in the disease state, and does it contribute to the pathology of the disease? And the answer is clearly yes and yes for Tie2. Tie2 activation is a fundamental mechanism which stabilizes vasculature. It keeps the support cells intact and needs to be operative to have healthy vasculature throughout the body. We know that Tie2 is inactivated in diabetic vasculature, and one of the hallmarks of diabetic vasculature is loss of parasite coverage. We know that Tie2 contributes to that, and it contributes to endothelial cell degeneration. So clearly the mechanism is in play and contributes to the pathology of the disease. Now, what we have exploited is there's a negative regulator, a protein tyrosine phosphatase called VE-PTP, which is upregulated in diabetic vasculature. It's upregulated in the back of the eye, as well as the vasculature throughout the body of a diabetic. This VE-PTP is the master switch that shuts off Tie2. So we know it's also operating in the disease state in the vasculature of interest. Now, what we've done is we've developed a small molecule inhibitor of the phosphatase. And when you inhibit that phosphatase, you remove the brake on the system and you activate Tie2. So we know this in cell-based assays, we know this in animal models, and we now have efficacy in humans with this mechanism. What this will allow us to do is be the first patient self-administered drug via subcutaneous injections that can treat both eyes and has the potential benefit of treating other diabetic comorbidities like the kidney disease and the other problems in peripheral vasculature. We do have clinical POC in patients with diabetic eye disease, and we are excited to be running a Phase IIb trial, which I'll tell you about momentarily. The clinical proof of concept we achieved in our Time 2 trial. This data was published and presented previously. This was a 3-month study in patients with DME where we had 9778 monotherapy. We had a combination arm with Lucentis as well as a Lucentis monotherapy arm. And in a pre-specified analysis, we actually got a signal of efficacy for diabetic retinopathy in the monotherapy arms. What was exciting to us is we got about a 10% number of improvers and 2-step improvement from baseline in both the study eye, and we also saw it in the fellow eye. And in the study eye, we got a comparable effect to Lucentis, and in the fellow eye, which didn't get treated with Lucentis, we were basically more effective than placebo. So this led us to believe that this could be a really game-changing therapy for earlier stage disease where we could go after non-proliferative diabetic retinopathy. Also in that trial, and also published, was the endpoint for DME in the study eye. And here you can see that the combination therapy of Lucentis plus 9778 was much more effective at reducing retinal thickness in these patients, and this was a highly statistically significant result. So our development strategy is to bring 9778, the small molecule, forward for non-proliferative diabetic retinopathy, where there's really no established treatment paradigm, and there is an established regulatory pathway. And we are bringing the large molecule antibody to VE-PTP forward for combination therapy with the anti-VEGFs for the more serious disease, DME. Now, why diabetic retinopathy? Obviously, there's 1 in 3 patients out there that have diabetic retinopathy, but only 1 in 15 progress to DME. So the market potential is potentially much larger for diabetic retinopathy than it is for DME. So what we are embarking on now, and have already started, is a 1-year duration trial in non-proliferative diabetic retinopathy patients. We are randomizing 150 patients into 3 arms, 1 to 1 to 1. One will be a BID group of 15 milligrams 9778 BID. One will be a QD dosing arm, and the third will be placebo-controlled. The primary endpoint, of course, will be the 2-step improvement at 1 year in the diabetic retinopathy score. This is an established regulatory path, and this is essentially the approvable endpoint that we would hope to achieve in this Phase IIb study. So we do have the potential to transform diabetic eye disease. 9778 will be the only patient self-administered product that will be used in non-proliferative diabetic retinopathy. Again, we think it's the least invasive and potentially the most effective way to activate Tie2. And we know the Tie2 mechanism is clearly in play in the disease state, in the back of the eye, as well as throughout the body in diabetics. So we also hold global rights to this, and we are very excited to be starting the trial and moving forward with the program. So thank you all for your attention this morning, and thanks to Emmett and the committee for inviting us again.